Welcome back to daily lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby, and we are on the subject of molecular biotechnology. With regard to the regulating aspects of the use of biotech, we're on to the specific topic of patenting biotechnology. The principal objective of biotechnology is to produce commercial products for economic gain. However, no company will initiate high-risk long-term projects without knowing that the results of its research efforts can be legally protected from competitors. At the same time, society at large has a stake in encouraging industrial innovation. A strategy that meets both of these objectives is for the government to grant inventors exclusive rights to the novel products or processes that they develop. Collectively, these sanctioned privileges are called intellectual property rights and include trade secrets, copyrights, trademarks, and patents. Trade secrets comprise private information about specific technical procedures and formulations that a company wishes to protect from others. Copyrights protect the authorship of published works from unauthorized use. Trademarks can be either words or symbols that identify a particular product or process of one company. For example, the term failsafe is the legally recognized designation for a polymerase chain reaction PCR procedure marketed by Epicenter Technologies. Other companies that sell similar PCR kits have created their own protected names. For biotechnology, patents are the most important form of intellectual property. A patent is a legal document that gives the patent holder exclusive rights to implement the described invention commercially. Moreover, on the basis of the extent of the claims of the patent, the patent holder can develop other products that are directly derived from the original invention, while competitors would have to license the right to use the invention in order to develop a product based on it. On the other hand, a patent is a public document that must contain a detailed description of the invention, so it informs others about the nature and limits of the invention, allowing them to decide whether they should continue working in a particular direction or try to use the patented invention as a springboard to other possible innovations. Patent decisions and laws vary from country to country. Although there are ongoing attempts to develop international standards, the duration of the exclusive rights of a patent is 20 years from the date that the application is filed in all countries. However, the United States specifies that if there is a dispute between a proprietor, the applicant who was first awarded the patent has the right for first 20 years first to invent principle. In almost all of the countries, the patent is given to the applicant who filed first. First to file principle usually takes 2 to 5 years following the filing of the initial patent application before a patent is granted. In every jurisdiction, the holding of a patent can be of considerable economic value, and it is not a trivial matter to receive one. For this reason, both the patent application and the invention must meet a very strict set of criteria. Generally, for either product or process to be patentable, it must satisfy 4 fundamental requirements. The invention, after having been shown to work, reduced to practice, must be novel, meaning that the invention does not exist as another patent that is held by someone else in another country, is not an existing product or process, and outside the United States has not appeared in some published form before the submission of the patent application. In the United States, an inventor has 1 year following publication in which to apply for a patent. A patent cannot be granted for something that was merely previously unknown, that is, a discovery, rather than the invention must contain, as judged by the patent office, an inventive step that was not obvious to other workers in the field. The invention must be useful in some way, whether it is a process, an instrument, a compound, a microorganism, or a multicellular organism. Every patent application must contain a description of the invention that is sufficiently thorough that a person knowledgeable in the same field can implement it. A patent cannot be granted for anything that is a product of nature. The notion here is that it is not appropriate for society to give a monopoly to someone for something that occurs naturally, has merely been discovered, and therefore belongs to the public. Often companies and individuals skirt this constraint by applying for a patent that covers the process of purification of a product, thereby avoiding the direct question of ownership of either a natural substance or an organism that produces the product. In some countries, such as the United States, according to the Supreme Court in a landmark decision, virtually anything under the sun that is made by man is patentable. However, in other countries, including members of the European Union, therapeutic and diagnostic procedures are not patentable. There is no simple, immediate system for granting the patent or for granting of a patent. The application must be prepared by an expert, normally a patent lawyer, and is organized in a defined pattern. In the United States, the application must have a title and abstract describing succinctly the nature of the application, a section on the background of the invention that includes a full and open description of the current state of the art and the field of the invention, a comprehensive summary of the invention with, if considered helpful, figures and schematic representation sections that explain the nature of the invention and describe how the invention works, and finally a list of claims about the invention and how the invention may be used. The application is sent to the U.S. Patent and Trademark Office, PDO, where it is reviewed by an examiner for novelty, non-obviousness, utility, feasibility, and general acceptability as a patentable invention. And in the United States, the application must have a title, an abstract describing succinctly the nature of the application, a section on the background of the invention that includes a full and open description of the current state of the art in the field of invention, a comprehensive summary of the invention with, if considered helpful, figures and schematic representations, sections that explain the nature of the invention and describe how the invention works. And finally, a list of claims about the invention and how the invention may be used. The application is sent to the U.S. Patent Office and Trademark Office, PDO, where it is reviewed by the examiner for novelty, non-obviousness, utility, feasibility, and general acceptability. When an examiner agrees that the invention meets all criteria for patentability, then the patent is awarded. However, the receipt of a patent is not a license to produce and sell the invention. All statutory regulations must be met before any product can be marketed. For example, if a patent is granted for a genetically engineered microorganism, the manufacturer must satisfy the recombinant DNA regulations for its production, distribution, and release. Protection of patent rights is the responsibility of the patent holder, and generally means that that means bringing a lawsuit against those who are presumed to be infringing on the patent. These disputes are decided by the courts and not the patent office. Similarly, if a person or company feels that an awarded patent is inappropriate, the legitimacy of the patent can be challenged by a lawsuit. If a patent application is rejected by the examiner, then the applicant can appeal the decision of a patent appeals board. If this appeal is turned down, then the decision can be challenged legally. For some applicants, patenting can be a frustrating experience. There are a number of cases in which the stakes are considered to be so high that costly court battles go on for years. Thomas Edison, who held more than 1,000 patents in his lifetime, once described a patent as an invitation to a lawsuit. Product patents and process patents make up the two major categories of patents. Products include homogeneous substances, complex mixtures, and various devices. Processes include proprietary procedures and methodologies or actual uses. The patenting of biotechnology innovation has been based on the historical experience of patenting inventions with agricultural, fermentation, pharmaceutical, and medical industries. For example, in 1873, Louis Pasteur received 2 patents, US patents 3525 and other 141072 for a process for fermenting beer that included the living organism yeast used in the process. Today, most but not all biotechnology patent applications are straightforward and patents are granted without any significant problems. Today, most but not all biotechnology patent applications are straightforward and the patents are granted without any significant problems. However, the first time a scientist attempted to patent a genetically modified microorganisms that was engineered by the introduction of different plasmids, each of which carried the genes of a separate hydrocarbon degraded pathway, the case was highly controversial. The genetically modified bacterium, which is capable of breaking down many of the components of crude oil, was developed by H. Chakrabarty, who at the time worked for the General Electric Corporation (GEC). However, despite its potential usefulness in cleaning up oil spills, the patent application of the bacterium was rejected by the USPTO on the grounds that microorganisms are products of nature and as living things are not patentable. However, in 1980, a landmark decision made by the US Supreme Court decided that this organism was patentable according to the US Patent Statute, arguing that a live human-made microorganism is patentable subject matter as a manufacture or composition of matter. The argument against patenting this genetically engineered microorganism tended to center on how the organism was developed in the past. Induced mutation followed by selection of the novel properties was an accepted way to create a patentable living organism. However, genetic engineering was considered by some to be tampering with nature. Consequently, it was argued that no inventor should benefit from manipulating products of nature. This position was not upheld. Thus, in the United States from 1980 onward, and later in other countries, organisms, regardless of the means that were used to develop them, must be judged by the standard criteria of novelty, non-obviousness, and utility to determine if they are patentable. Patenting in different countries. The rights given by a patent extend only throughout the country in which the application was filed. Therefore, to protect an invention, a patent application must be filed separately in each country. And although the World Intellectual Property Organization, WIPO, is attempting to develop international standards, patent offices in different countries often reach quite different conclusions about the same patent application. For example, in 1989, the biotechnology company Genentech applied for a patent in the United Kingdom for, among other things, the production of human tissue plasminogen-activated TPA by recombinant DNA processes. These proteins exist in small amounts in the human body and converts plasminogen to plasmid. Plasmid is an active enzyme that degrades a fibrin of a blood clot. Consequently, human TPA has been considered as a possible therapeutic agent for the prevention and treatment of coronary thrombosis. Genentech, after considerable effort, assembled a complete version of a human TPA complementary DNA, cDNA, and cloned this cDNA into E. coli for the production of large amounts of pure TPA. In its patent application, Genentech claimed rights to human TPA as a product based on certain procedures of recombinant DNA technology that they developed, the cloning vector system, and the transformed microorganism as a part of the process. Categorized category protection for the use of human TPA as a pharmaceutical agent was also sought by Genentech. A total of 20 claims were presented in the original patent application. Some of these were broad and others were narrow in scope. The patent was rejected by the United Kingdom's Patent Office. Genentech then appealed to the United Kingdom's Court of Appeals, which after considerable deliberation invalidated all claims for a variety of reasons. The judgment concluded that the patent was novel, but some of the judges argued that the submission was obvious, therefore it could not be patented. In contrast, Genentech was readily awarded a patent for human TPA. In the United States, the US patent not only protects the form of human TPA that was to be marketed by Genentech, but also gives Genentech exclusive rights to all similar but not identical active forms of human TPA. Genentech won a lawsuit against two other biotech companies that were found to be infringing on its TPA patent, although they were selling non-identical forms of TPA. The Japanese version of Genentech's TPA patent is limited to the amino acid sequence of the human TPA that was cloned and patented by Genentech in Japan. Other companies can sell variant forms of human TPA. Thus, basically, the same patent application was rejected, approved, and given a broad interpretation, and approved and given a narrow interpretation by 3 different patent offices. For the present, at least, there are divergent views about what is or is not a patentable invention. Patenting DNA sequences. Patenting DNA sequences. Currently, isolated nucleic acid sequences, whether DNA, RNA, or cDNA, derived from RNA and proteins, are patentable. All the gene sequences and the mRNA and proteins encoded in these sequences are found naturally in organisms. Purifications from their natural state is considered sufficient to render them patentable. Since 1980, thousands of patent applications for whole genes have been approved by patent offices throughout the world. The United States, more than 40,000 DNA-related patents have been issued at a rate of 3,000 annually since 1998. Almost 20% of human genes have been patented. Some of these genes are used to make therapeutic proteins such as recombinant erythropoietin. Erythropoietin stimulates the formation of red blood cells and is used to prevent anemia in patients with kidney failure who require dialysis. Many of the other patented gene sequences are used as diagnostic probes. One example results from the discovery that particular mutations in the human gene BRCA1 are linked to breast cancer. A patent issued by Myriad Genetics Incorporated claims methods to detect these mutations in BRCA1 to diagnose a predisposition to breast cancer. With the rapid accumulation of genetic sequences from genome sequencing projects and the undertaking of the partial sequencing of thousands of cDNA molecules from different organisms, tissues, and organs, the patenting of nucleic acid sequences became extremely contentious. In 1991, the issuing of patenting gene frames was broached when scientists from the United States National Institutes of Health filed for the patent rates of 315 partially sequenced human cDNAs expressly sequence tags ESTS. 2 additional filings brought under the total number of partial sequencing of 6,869. In 94, in a preliminary ruling, the USPTO notified the National Institutes of Health that it would reject the patent application on the grounds that the functions of the sequences were not known. In other words, partial sequences by themselves did not fulfill the requirements of utility and were not patentable. However, by 1997, over 350 patent applications for more than 500,000 partial DNA sequences had been filed, mostly by private companies, which purportedly met the standard for usefulness. One of these patent proposals sought protection for about 18,500 ESTs. Consequently, serious concerns were raised about granting patents for large numbers of sequenced genes and partially sequenced DNA fragments with broadly based applications. Individuals who oppose the patenting of DNA fragments with unknown or loosely defined functions contended that genes and partial DNA sequences or discoveries are more likely products of nature and definitely not inventions. Others conceded that although eventually some of these sequences might be useful, it was premature and speculative to award patents without additional information about the functions of the sequences. In this context, the thousands of ESTs are considered to be means to ends and not the actual endpoints. On the other side, those who favored patenting ESTs maintained that these collections were novel because they defined the novel messenger RNA, mRNA complements of various tissues and organs, and consequently had utility because each collection could be used as a diagnostic assay to determine the extent to which a disease alters the normal complement of mRNAs in various organs. After developing some ad hoc rules, the USPTO examined in more detail a full range of its use and concluded that genes and partial DNA sequences were patentable. On Jan 5, 2001, a set of guidelines for gene patenting was released. The key requirements for this type of application was that each DNA sequence must have specific and substantial credible utility. Moreover, the written specifications and claims for each sequence must be thorough and demonstrate the actual use of each sequence and not merely a potential function. These guidelines have established the criteria for patenting incomplete DNA sequences in the US. Although the granting of patents for any human DNA sequence remains controversial. And also controversial is the increased scope of the DNA patent claims in recent years, often included in the claims to DNA sequences. And the protein is then encode are antibodies against the protein, even when the antibody has not actually been produced. Antibodies against human proteins are important from a commercial perspective because they are used for diagnostic purposes and as therapeutic agents, and therefore there is considerable incentive to include them in patent applications. However, some argue that they do not meet the criteria for patentability because in many cases the antibodies have not been produced and therefore the characteristics are not specifically described and working examples are not provided. Patenting multicellular organisms. The patenting of multicellular organisms continues to raise ethical and social concerns. However, there is nothing intrinsically new about the exclusive ownership of living material. In the past, microorganisms were routinely patented and specific laws were promulgated to give plants breeders the right to own various plant varieties. The transgenic mouse, onca mouse that carries an activatable gene that makes it susceptible to tumor formation, has been the precedent-setting case in many jurisdictions to determine whether genetically modified animals are patentable. Currently, patenting of genetically modified animals is sanctioned in most developing countries, including the United States, members of the European Union, Japan, Australia, and New Zealand. However, in other countries such as Canada, it is still not possible to patent a multicellular organism such as the onca mouse. Vigorous challenges to patenting transgenic animals that have been put forward on moral grounds. In other words, the issue is whether society considers this form of patenting acceptable. From a historic perspective, it is unlikely that a position based on ethical consideration will be completely successful in preventing the patenting of all transgenic animals. However, patenting is not an absolute right, and governments, by passing specific laws, can determine what can or cannot be patented. If an invention is considered by various special interests groups to have potentially negative economic impact on existing agriculture practice, for example, then it is quite possible that a law preventing the implementation of the new technology could be passed. On to the milestone note. On transgenic non-human mammals, inventors, courtesy and gratitude to P. Leder and T.S. Stewart, assignee president and fellowship of Harvard College, Cambridge, MA. And so U.S. Patents 473686612, April 98. In 1980, the US Supreme Court defined a patentable invention as one that included anything under the sun that is made by man. In 1988, a transgenic mouse was the first genetically engineered animal to be patented. In this case, the transgene considered of a cancer-causing gene, oncogene, driven by a promoter in the long-term and repeated mouse mammary tumor virus, MMTV LTR. The oncogene was the MYC gene from the chicken myelocytomatosis, OK10 virus. The invention entailed cloning an MMTV LTR MYC fusion gene into a plasmid, injecting linearized plasmid DNA into the male pronuclei of fertilized one-celled mouse eggs, identifying offspring that express the MYC gene, and establishing transgenic mouse lines. In some of these lines, the MYC gene was expressed in several different tissues, and in other lines, it was limited to one or a few tissues. The integration of the MMTV LTR MYC gene construct, according to Leder and Stewart, increases the probability of the development of neoplasms, especially particularly malignant tumors in the animal. These transgenic organisms can be used to test whether a compound either causes or prevents cancer, and as a source of cell lines from cells of various tissues, such as the heart, that are difficult to culture. Since 1989, DuPont has been selling one of these lines of transgenic mice under the trade name OncoMouse. More generically, others prefer to call this mouse line the Harvard OncoMouse, or for short, just OncoMouse. The granting of U.S. patent 4736866 was contentious, with much of the concern directed at the ethical implications of such patents. Those who oppose the patenting of transgenic animals argue that this type of patent violates the sanctity of life, threatens the integrity of species, and fosters inhuman treatment of animals. Despite these allegations, since 1988, hundreds of patents have been granted in the United States for various transgenic organisms. For example, there are now patents for transgenic animals that act as models for benign prostatic disease, inflammatory disease, altered fat tissue, metabolism, and thrombocytopenia, to name a few to date. Neither the U.S. courts nor the U.S. government has suggested that in principle any of these patents is inappropriate. The parenting of transgenic organisms is no longer an issue in the United States, and after much discussion and litigation, transgenic animals are now patentable in most jurisdictions throughout the world. Vigorous challenges to patenting transgenic animals have been put forward on moral grounds. In other words, the issue is for whether society can go ahead with that or not. From a historic perspective, it's unlikely that a position based on ethical consideration will be given completely successful in preventing patenting of all transgenic animals. Patenting and fundamental research. Not everyone believes patenting is worthwhile. Some opponents argue that awarding a monopoly restricts competition, leads to higher prices, curtails new inventions, and favors large corporations at the expense of individual inventors or small companies. Despite these concerns, the patent system is well established and is here to stay. Moreover, patent ownership does not appear to prevent significant research and development by other researchers and companies. Indeed, it might be argued that if patents were serious impediments to innovations, then U.S. Patent 4237224, which was granted to Stanley Cohen and Herbert Boy in 1984 recombinant DNA technology for both the use of viral and plasmid vectors and the cloning of foreign ones, should have seriously constrained the development of recombinant DNA technology. This lecture will be over in a couple of seconds from now. We'll be continuing in the next lecture. Obviously, no such hindrance has occurred in the past. Patenting and patent enforcement were rarely of interest to academics, researchers working in the biological sciences. Now, however, there is a view within the academic scientific community that patents and consequence of patenting may be detrimental to established scientific values. Traditionally, science, especially university-based research, has been an open system with free exchange ideas and materials through publication and personal communications. We'll meet in the next lecture. Take notes, screenshots, and prepare well. Thank you. Best wishes.